So I'm Irv Weissman from Stanford, and while my slide, or slides, are getting set up, I think I'll do something strange. I'll start off by saying the definition of a stem cell. So we're halfway through the meeting. Might as well get to it. So the definition of a stem cell is a cell which, at the single cell level, when it divides to give rise to two daughter cells, on average, one of the two is at the same stage of development. That's called self-renewal. The property of self-renewal in all tissues that have stem cells is limited to stem cells. So in the blood-forming system, for example, here we go. I had it, there it is. The stem cell, this rare cell, is only 1 in 20,000 cells in the blood-forming tissue, and the blood-forming tissue could be bone marrow or mobilized peripheral blood or umbilical cord blood. So only 1 in 20,000 is appropriately called a stem cell. And now you could ask, and I will ask in a minute, how many stem cell therapies really exist? Now we found back in 1988 to 1992 how to isolate blood-forming stem cells pure. And the practice of medicine then, unregulated, well, sort of unregulated, was to take mobilized blood or umbilical cord blood, thanks to John Wagner, or bone marrow and transplant it. Now, as you can see the way I've drawn it, there are a lot of other cells in there besides stem cells. Yet stem cells are the only cells in the blood-forming system that will regenerate the blood-forming system, and they only regenerate blood and no other tissue. I've tested it. I've tested it with mouse. I've tested it with human. I've given it every possible advantage. I had co-founded a company about isolating and transplanting stem cells, so I had a huge financial interest for it to be able to regenerate all tissues of the body. But it didn't. When you give an autologous transplant person back to itself, it's always to treat a cancer. It's so that you can give a higher dose of anti-cancer drugs than you could give before. But as we showed, in metastatic breast cancer, non-Hodgkin's lymphoma, and we didn't show it so well, in myeloma, giving cancer-free stem cells, because you purified them, is better than giving a bone marrow or a mobilized blood transplant that's cancer-containing. And that's statistically in progression-free survival and overall survival at all time points. Yet it's not a therapy. We showed that 1996 to 2000, and it's not a therapy, which is another barrier we hadn't thought of. We had done this so that we could change the treatment of people. But we were bought by a company, very friendly company, that said, and for a lot of money, that said, we want to join with you and take it forward. Within 4 years, their leadership had changed as they merged with another big company, and this wasn't going to make enough money in 5 years to continue it, so they shut it down but kept it. You know what I mean by keeping it. Okay? And we were also going to transplant healthy stem cells from a donor into somebody who had a genetic blood disease. Shut down, hasn't happened. So if you talk about barriers, one of the biggest is the companies that were itching to get into this field but didn't have any idea of what the field was. Next. Next. There we go. So before we isolated stem cells, and by the way, I think we're the only ones who have ever transplanted pure stem cells. Blood forming back in the 90s, and then brain forming from another company. So hardly anybody actually transplants just stem cells. So before 1988, the bone marrow transplant group called what they did bone marrow transplantation or mobilized peripheral blood. After the name stem cell got into the community, without changing what they did, they said, we all give stem cell transplants. And that confuses people, especially oncologists, because they're the bunch of pre-meds who got straight A's and could memorize their way through school. I know I'm going to piss everybody off, but it's okay. They could memorize everything, but they didn't know where the information was obtained. And no matter what I've done, and I talked to ASCO and American Society Associated in Cancer Research, I can convince anybody it's safer to give cancer-free stem cells than cancer-contaminated stem cells, except oncologists and a lot of bone marrow transplanters. So this is a big, big problem. Next. So the next problem, when we wanted to move forward on all kinds of stem cell research, I was the head of an NASIOM panel on reprogramming stem cells for therapeutic use, was immediately everybody came in saying, everybody including from my own university, saying that blood-forming stem cells, when they pass through the heart, turn into heart stem cells. When they pass through the lung, they turn into lung stem cells. When they pass through skeletal muscle, they make skeletal muscle stem cells. When they pass through the brain, they turn into that. And that was picked up by the Vatican and the Church and other fundamentalists to say, why are you talking about making embryonic stem cells and killing embryos when we know already? Now, Rusty Gage and I reviewed at least 2 of the papers. We rejected it in major journals, but we were overruled by the editor alone because it was very sexy to say blood makes brain, blood makes liver, and so on. So you've got to watch out for what the evidence really says. There is no transdifferentiation. We cannot find in the body of living people any source of pluripotent cells capable of making all of these except by genetic modification. Next. So when they got through with all this, I had to say to them, hey, look, what are the generally accepted requirements to establish that a discovery is a scientifically acceptable fact? Well, first, it's got to be published in a peer-reviewed journal. But those of us who are scientists know that's clearly not enough. The recent article by Glenn Begley says only 30% of papers published in those 2 major journals can be reproduced. So it has to be replicated in several independent laboratories. But I think that's not even enough because a lot of people can fool themselves about what the date is. However you look at the principle that's being revealed, this story has to come out. If one kind of stem cell in one tissue turns into another, it should always be revealed no matter how you look at it. But here's the important one and what we're talking about today. If you're going to treat people in regenerative medicine, you have to have robust, rapid, and sustained regeneration. And I put to you that hardly anything has met those criteria that are in the clinic today, at least as purified cell products. Next, please, because I promised I'd get done before 10 minutes. So at the same time, because probably John Wagner and the others were doing so well with umbilical cords, the umbilical cord companies and others said, well, look, they're pluripotent in the umbilical cord. And so we tested both umbilical cord blood and purified stem cells in a model where any tissue stem cell from a human grows in the mouse. So we're actually doing the human stuff. The next slide. I might have the numbers. This is just to show. This is the percentage of cells of those tissues made by the umbilical cord. This is the amount of blood that was made in the mouse and easily detected. So there is no evidence whatsoever that a cell can normally transdifferentiate from one fate to another, much less become another tissue stem cell. Next. Here's where I'll get in trouble. The field of mesenchymal stem cells began actually probably with Arthur Kaplan. Arthur or Arnold? Arnold. Arnold. Arthur's a different yes. Wow. You don't want to get on the wrong side of him. But Arnold Kaplan was working on bone regeneration and non-healing fractures and came up with bone-derived cells, stromal cells that could make bone cartilage, maybe fat. But when you look at the mesenchymal stromal cells that are grown from bone marrow, oh, look at that. There it is. There is no evidence of the presence or frequency of self-renewing cells. So they're cultured bone marrow cells. And it may be 1% or 0.1%. I don't think it's 100%. So whatever is happening with mesenchymal stem cells is really the property of mesenchymal stromal cells because they don't self-renew and they don't regenerate as part of their outcome. Fine, perhaps, as immunomodulatory treatments, but that's black box science that needs to be revealed at some point. But of the proven properties of what's out there as mesenchymal stem cells, they are not being used to regenerate, and so they are not being used as a stem cell. Next. Should be almost the last. Oh, yeah. So as time went on, a group in Louisville, headed by Ratajak, claimed that we all missed the stem cells because they were so tiny we would never get them out of our cell sorting. So he called them very small embryonic-like stem cells, formed a company called Neostem, backed principally at the beginning by the Vatican. 2 meetings between the Vatican and Neostem, huge meetings, proclaiming these as moral stem cells. Yet, when we went back and looked for every property, you can read it, it's in stem cell reports, they don't exist in the mouse as reported. We can't find any pluripotency. We can't even find, in that fraction, as they claim, blood formation capacity. Next one. So this and other things led us to what you already heard today many times. What are the real steps to a clinical therapy? Discovery that has to be independently replicated, preclinical and published proof of principle, application to national other body for successive trials to demonstrate safety, efficacy, and approval from the body that the trial was safe and efficacious. IRBs don't do that. IRBs oversee the clinical trial while it's going on. Next slide and last slide. So at ISSCR, in a closer look at stem cells, we proposed 2 simple questions for somebody who is a caregiver or a patient looking for stem cell. Ask the provider to provide the IRB protocol and the name of the head of the person of the IRB that oversaw the trial during its clinical trial, and we would then be able to find out if it was independent or rather dependent, as I hinted at. And the second is, show us the evidence that your state or country regulatory body equivalent to the FDA has shown it's safe and efficacious. That's all we did. And Nancy Whitty and Heather Rook can tell you the response. 500,000 patients on the 500,000 hits on our website, a letter from a lawyer in a particular city in the United States saying, by what authority you're asking the questions, and the fear of litigation led it to be shut down. Well, that society is a poor society, and it doesn't have the power to promulgate the kinds of things we need to do. It's too much to put on a single society. So hopefully today, yes, in the morning and the rest of the day, we're going to be able to figure out what's the best way to get there and how do we protect people while still allowing capitalism to succeed. Michael Felland is a patient. Come on up, Michael. And he's going to tell us about his experiences in cruising the stem cell field.